full wide.
Think you're moving yet, Copilot?
Starting to.
So this is just a local high right here that I'm on top of.
So current depth is 2,315 meters. We're diving on a ridge to the south of Pioneer Bank, up in the northwestern Hawaiian Islands, part of the Papahanaumokuakea National Monument.
Kind of looking like we have reached another local high. Again, these local highs located along this ridge are an indication that we have actually located an old volcanic vent that was generating these lava flows that we've been diving on currently. And the reason that we see so many active lava, or not active, excuse me, so many lava flows on these ridges is the rift zone ridges are the locus of many of the volcanic eruptions that occur in Hawaii. You can equate what we're diving on right now as to something that is located in the Big Island of Hawaii, where Pu'u'o'o is just down the rift zone ridge from Kilauea.
Is that a sea star on the left, on the back?
Yes.
Let's take a look. I'm sure there's some interest.
Okay, video. Let's go in on this sea star.
Roger, coming in. And holding.
Okay, centering up.
Coming in a little bit more. Holding. A little bit more.
Yeah, let's hold that till you're done and then we can go in.
Yeah. Got it there.
Copy that, Bridge. Thank you. Move is complete.
So I don't know if Chris Ma might be watching this from the Smithsonian Institution.
Want me to tilt down to the tip of an arm?
Yeah, let me come out just a little bit.
Sea stars and echinoderms.
I wonder what's propping this guy up too.
Oh, he's on top of something.
Consuming something on the backside?
Yeah, I could rotate around there. Actually, let's go on his feet down here first.
The closest I can come to is that it's in the genus Evoplasoma,
holding,
the family Gonasteridae. But it looks a little bit different on the images I have here in my gallery.
Yeah, it's like a little white coral of some sort behind him.
Yeah, sure he's in this position where he's off the bottom for a reason.
So sea stars are
okay, once
carnivores.
You're clear from that,
they feed on
you can go wide and I'll reposition around the other side.
Coming out.
Predators are also considered keystone species, having a very disproportionate effect on the ecosystem based on their abundance alone. So not being very abundant, but very important components of the community. if we can. Are we still waiting for a ship move? Or have some time in this
no,
area, or are we moving?
I was just going to get the backside, and
Okay.
then we can move on. Sirius is
Perfect.
moving, yep.
Thanks.
It looks like he's eating something, so. Okay, video, let's do this one while I'm floating here.
Roger, let's try this. Holding there.
So yeah, there you can see exactly what I was talking about these sea
Coming up.
stars being voracious
You
predators.
come
of,
up, or we could just tilt up.
corals,
This seems like a good
sponges,
angle.
and other
We just got
invertebrates.
the camera up a little bit.
Look like it's feeding on a
Yeah, I'm going to come in
and
some more if you're
presumably
okay.
Yeah,
Roger that.
primnoid or
There we
bamboo
go.
coral.
coming up.
I'm just going to hold the zoom. It feels like
Okay.
you're going to come up.
Yeah.
So yeah, beautiful shot there. And quite appropriate. We're all just rotating through lunch here on the ship. So and yeah, Scott is just noting that this sea star is feeding by averting its stomach around the coral.
Similar to our lunch.
Beautiful imagery.
Great. Okay, video. Once you're done your shot,
Roger.
move on.
Co-pilot, is Sirius still moving?
I think we're slowly coming to a stop, but I have no good feel for which way the ridge continues. Looks like it would be a little more north now, but
It should be generally north. I can try to face my sonar that way. Yeah, we're at a local high, so it's
looks like it is off to the north a little bit, but hard to tell.
So just updating everyone on
definitely not
position.
this
We're
way.
about a third of the way
Yes, you
of
can
what
see
we're
it on
hoping to
sonar.
accomplish on today's dive.
It's off that direction. Can you give me a heading of that strike?
turning.
Yes. From the sonar screen, or do you want me to look?
He's just going to line up, put it directly
Current,
center, and
yeah,
then
has
we'll look
been
at his heading.
Very mild
Okay.
to
So it's about
non-existent
15.
on this ridge crest.
Yeah, 15.
So that's your next
The
move.
only indication that there is current up
Copy
here
that.
has been,
Another 30 meters.
Yep. Let's do
from
another
the
30
sand
meters
ripples we saw
due
at
north.
the beginning of the dive.
Okay.
Because
You can see it's
need to
kind of
face
very
off that way.
slow. The particles
Copy
in
that.
the water column coming from right to left, so that
Bridge,
would
this
be
is
from
nav.
east to west.
We'd like to request another ship move. 30 meters at due north, 0.2 knots. Good copy.
If we have a second, there's a sponge to the lower portion of this screen right in front of that boulder.
Okay, between us and the boulder?
Correct.
Gotcha.
Copy that, bridge.
Just coming around to see the opening here. I think I'm kind of on my tether though.
you're tugging me.
So
Yeah.
if it's difficult, it's I know what it is. So that's in the genus Poliopogon, and that's species B.
We are heading that way, though.
Yeah. We are headed this way. I'll just pause for a second.
Okay.
Wait till Sirius comes to me.
Just wanted to check on our shore-based scientists at several of their exploration command centers, as well as those listening on the conference line. If you can confirm that you can all hear us still.
Maybe.
One point I'll make here is that as you see that
Can you come down a little, co-pilot?
I'd rather not. I have that big local high.
Oh, just below
Boulder
you.
right, kind of below me.
Okay.
I don't think my altimeter's showing it, but I think it's on the front end of my vehicle.
Oh, so you're not to it yet. All right. Fair enough.
I mean, it's kind of right down here. I'm sure I can see it if I bring my camera down.
so yeah, one point I'd like to make
I
here is
can
that
open
as
you
you
up a
see
little,
those
co-pilot, if you're
particles in
looking
the
for
water
it. A little
column
bit.
going from right to left or from east to west in this case.
Might be a
so
little bit
that's
to starboard.
the prominent current
Yeah,
direction.
it's off somewhere over
You
here.
can see that all the sponges and corals are also all oriented into that
I
same
would just
direction.
back up, pilot, and look
So
at
being
something closer to
oriented
me for a while.
perpendicular to their current,
That's where
so
I'm headed.
the fans and as well as those corals that have the sponges that have large surfaces, they're all oriented in a way that they face the current flow, and that's of course so they can maximize their feeding efficiency. So they feed on those tiny particles, plankton that's dissolved in the water column.
Ooh, there's a swimming sea cucumber.
Oh, spectacular.
See the magic.
Okay, video. Let's start going in.
Roger, going in.
Okay, hold there.
Holding. Gotta get focus on this guy.
So we're
Really
looking
hard to
at
tell
a
how close he is.
sea
Yeah, he's
cucumber
coming close.
It's
swimming.
coming at me.
He's coming right
Beautiful
at you.
imagery.
Oh, he doesn't have the black background anymore. Can we get the lasers off, co-pilot?
I think he's gonna come to port.
Yeah. Oh, there we go.
There's your black. Come on. Alright. Go slowly
can
brighten
go-
him up.
Can go in a little bit more.
Yeah, absolutely
Yes.
spectacular scenery. So yeah, most people know sea cucumbers from just these motionless creatures that lie on the seafloor.
Can we get a little bit more zoom video?
Roger, coming in.
This one
That's
will
good
be
there.
hard to
There,
ID.
yeah.
There are two families. And that could possibly be in the Elpetidae or the Cycropatidae. Both of these have these translucent.
Okay,
or
how should we
have
end
species
this?
with these translucent.
We should wag to
bodies.
starboard and let him go off to
Another
frame left.
interesting thing
Okay.
about sea cucumbers, there's a lot of species, including some in shallow water, that are used for food
Yeah,
and
good.
for
Come
medicinal
on.
purposes.
Alright,
So in a
thank
lot of
Pull
Asian
wide.
cultures, sea cucumbers
Was that
are
magic,
considered a
co-pilot?
delicacy and used also for medicinal
Nav, I
purposes.
think we can do another 30 due north.
Another interesting thing about sea cucumbers
Copy
is
that.
they
Try
can
to beat
actually
the weight this time.
change the
Copy that. I think they're
consistency
just
of
about
their
done moving
body.
anyway, so I'll
They
just
have
tell
this
them
Yeah,
to.
look like
collagen
they're
in
on
their-
target.
Yeah.
in their skin, so they can adjust the shape to fit through very narrow cracks or
Bridges, Nav.
kind of mold themselves as ever they.
Could we go
however
ahead and
they need
continue
to.
this move with another 30 meters due north at 0.2 knots? Good copy. Thank you.
So yeah, the large sponge there kind of looks like the half of an orange. That's another furriid sponge, and the genus Poliopogon. And that's currently known only as Poliopogon species B, so there's at least a dozen species that we know of here in Hawaii. And one of the ways that we use to differentiate them is dependent on how many of these atrial
Copy that, Rich.
surfaces
Thanks.
they have. So this one has a single atrial or surface there on the top, and we're looking up right now. And actually, there's a red coral right in front of us. Or was it bottom? So yeah, that is, as I mentioned, Poliopogon species B. So yeah, it's an anemone to our left.
Gotcha.
Let's take a look at that.
Are you done with this sponge?
Yep.
Okay. I see the anemone.
Pilot directly above that anemone, kind of hanging over the edge of that rock in the background is
And then
something
maybe
I
if we
can
can set up
identify.
also, like there's a request to zoom
After
in on
this.
that white stuff
Okay.
on the boulder behind it. So,
All right, video. Let's get this
Coming in.
anemone.
Holding,
Okay, tilting down.
so that's
coming in,
an anemone
holding,
in the genus EX1603. So this genus has both brown and orange species. It's one of the most
Really
conspicuous
opening up.
sponges here in Hawaii. Anemones, of course, most people know more from shallow waters.
Bouncing
They're
down. Sorry
commonly
about that.
used in the aquarium trade. Interesting fact about these animals is they can actually move. So for those aquarium enthusiasts that have kept anemones in their aquarium, you can actually see them if you have the patience and time to devote to culturing these animals, is that they will detach from the substrate and then move and reattach. So yeah, I think so. There's a request to zoom on that white stuff on the boulder and just in the back.
Okay, can you go partial video?
Partial.
See if we can get it from here.
About there, maybe.
Okay.
Current depth is
Gotcha.
2,320
Pilot, will you
meters.
just tilt me up? Excellent.
Have we seen that thing on the upper left?
What are we going
before?
for? You're slipping back.
Yeah, hold on. Let's go wide. I'll
Come wide,
come back
full
up.
wide.
We'll just float by and look at it. Okay, you can zoom video.
Coming in, holding,
Coming up to it. Okay, you can go in.
coming in, holding.
And yeah, so the Tina had pointed this out, this white material on the rock.
Still partial.
We saw something
good.
similar earlier. Hard to see from this kind of.
Do you want to go in all the way?
Roger, coming all the way. Full zoom. In focus.
I still have your camera. If you want
Okay.
it back.
You can keep it for now. How we doing on the moves?
Good. I'm starting to come above you, but
Okay.
doesn't mean you have to move out.
Yeah, and that last move just
Feels like we
ended,
might be
so
drifting backwards a
So
little
just
bit.
wanted
Thanks,
a
video.
swing.
quick thing for our shore-based scientists that were
Okay, let's go wide
mentioning
video.
that I was
Yeah,
difficult to
wide.
hear, so we've adjusted the audio.
See
Scott,
that-
can you
oh.
now hear me better?
That thing's moving, isn't it?
What thing?
That large mass on the left, on the top. That is definitely moving. Oh, you're off bottom, right?
Yeah.
Hold on.
The white thing?
Yeah, do you see the right side of that fluttering?
No.
Oh, that could be noise. I'm going to bring my iris up for just a second just to check this count.
Okay.
Yeah, it's noise. Disregard.
All right. I'll continue on to the north.
So I am looking back at you, but there's nothing in our future that's going to speed you up.
So
needs to
just
speed you up.
coming out of view right now is another potential location of a volcanic vent. Again,
Thanks,
we
Bridge.
can kind of interpret these local highs as a place where a volcanic eruption once had occurred and focused itself. Some of the structures that indicated that there was active flow coming out of that were pillow lavas that seem to have flowed downwards from the local high.
Was the ship move 045?
Sorry?
Which direction was the ship move?
North, due north.
Due north. Okay.
Yeah, but now it looks like it's adjusted. Does 4-5 work for the next move? 0-4-5?
Well, we'll let pilot start to get out a little bit more, but yeah, we might need
Yeah,
to.
it might be. Oh yeah, I'm still below you. Okay, pushing uphill.
Camera's pointing straight down.
Okay.
So yeah, this is what we considered fairly moderate density. We are at a depth of 2,300 meters, so just about 1.5 miles deep, and 1,000 miles away from Honolulu. So we see several corals here in several groups. What appears to be the dominant groups at these depths are the chrysogorgid corals and black corals, as well as some primnoid corals. Also several sponges that we've seen in the area. Most of them hexactinellid sponges of several in the family Furidae,
Pulling you.
Yeah,
like this
that's fine,
enormous gramophone
just trying to
sponge that's coming
guess
into
what the
view
next
right
move's
now.
gonna be.
One of the most conspicuous inhabitants at these
Nav, I think
depths.
it's actually going to be 030 for our next move. Coming back to you, pilot. Sorry about that.
Okay, that's okay.
0-0.
And I'm going to go ahead and update our position.
Have we gotten nice zooms of this sponge?
Yeah, no need.
Okay.
So yeah, if we can like try to make, I'll point out stuff that's more, it's rare. Yeah, but if we can cover some ground, it would be a
Okay.
number one priority.
Let's do 030.
Are you ready for that now?
Wait, you're faced 100. Let's get back in line here.
I'm sort of cutting the corner here. You just follow the ridge.
Okay.
Coming up.
All right, pilot's ready for that move.
Yeah. Sounds good.
Let's make it 015, Sean. 015. Coming back down.
So I just updated our position on the Fledermaus, and we're about 400 meters away from where we landed. Current depth is 2,316 meters.
There's definitely a current up here. Always lateraling to starboard about 20 or 30%.
So yeah, pilot is just noting that we do have some current here coming from the east to the west, just going over the ridge here. If we have a second, maybe we'll just do a snap zoom on those corals. They're just the bottom of the screen.
Sure.
There seem to be several of these.
But I think we're going to end up repeating that move. That 015 at 30 meters. Once a year. When they get to their point.
So yeah, that's new for this dive. So that's another chrysogorgid coral. So they're kind of distinguished by these longer polyps, and so this species in particular, it's an undescribed species that we've been referring to as Chrysogorgia species sandalbags. So we have collected specimens of this in the past, and it's still an undescribed.
Here, if you want to zoom.
Roger.
Just on this
Going in
mass.
partial.
Okay.
So yeah, a lot of the species that we see down here are still undescribed, and so a lot of work to be done with these groups. So yeah, that's wonderful. Thank you.
All right. Go wide video.
Coming.
Thank you.
Wide.
Just to update all of you folks, shore side, we are about halfway into the dive. So we landed about 2.5 hours, and we still have about 2.5 hours on the bottom. We've covered just under 400 meters, and we hope to at least double that by the end of the dive, and then hopefully come into the area that was really high density. So there's another one of these chrysogorgid corals that's a branched, another species. And we're just hovering here for a couple of seconds. They're going through another personnel change with our pilots. Yeah, and Bruce made a very interesting observation here. Bruce Mundy from NOAA's Fisheries and Science Center, so that's a bit more noticeable, the density of corals and sponges. And yeah, so we've had a kind of a gradual increase throughout the dive. It's still somewhat moderate density, not really the super high densities that we saw last year and back in 2003. But it is an increase. Some of the most noticeable features are these massive pillow lavas, and then we've seen a lot of these dead sponge material kind of in between. And yeah, and Scott makes another interesting observation here, saying that a lot of the colonies appear to be small. So you can see a lot of these small. Most of them appear to be primnoids. So it appears to be a young community.
There you go.
Thank you.
So that a lot of times when those kinds of observations are made, that's linked to either a large recruitment event that happened in a particular point in time.
Copy that, Bridge. Thank you very much.
There's another one of these stocked sponges to our right.
Move is complete.
And that's
Copy that now.
very bizarre looking, and it's genus Bolosoma.
Lower swing arms are in.
Oh, they're out slightly.
Okay.
And so yeah, here we see a noticeable increase in the densities.
Let's do a snap zoom on this.
Roger, coming in.
So Dan, you mentioned that we're looking at a younger community of corals here. Can you give the public an estimate of maybe how old some of these corals might be?
So yeah, that's a very good question there, Jonathan. So for most of these things, we don't have growth rates. Very few of them we actually do. We do know that a lot of these corals are able to live centuries to millennia in some cases. I'm just going to try to look up this crinoid here. That's going to be in the
Full wide.
so that's a very noticeable. So that crinoid is an unstocked crinoid, so it's part of the group Comatulids, and it's Cerameta triceralis, and very certain of that. But back to the
30 meters, 066 degrees.
Yeah, back to the age discussion. So for a lot of these things, we don't know. Just a few species have been studied in the deep sea, and for those that have been studied, it's kind of a generality that most corals in the deep sea, as well as sponges, are very old. So in many cases, on the order of centuries.
Good copy. Thank you.
And that's another dead sponge. So here's a so yeah, dead sponges throughout. So when dead material is seen, that could be either some kind of that the environment is not very stable. There could be slides, but yeah, we've seen a lot of dead material.
Yeah, and Dan, you mentioned that there could be some small local landslides happening. We're looking at a talus field right now, so all these little boulders and rocks are very unstable. They're constantly moving downhill, and so that could be one reason why we're seeing a lot of dead skeleton sponges.
Yeah.
0045. I'm trying to take us back center.
So yeah, that's back to your age question. So corals, they don't have like other animals like we do, that we have programmed cell deaths, that at some point in our lives we basically our cells stop regenerating. Corals do not have that, and so if conditions are favorable, they could regenerate and continue growing. So a lot of times it's some external force that kills them, or in some cases, they just become too massive and basically tumble over, and that's something that we routinely see, that you see a very large colony that's just become too big for the boulder that's grown on, and then it basically just tumbles over, and some portions might continue to grow for a while.
What are you looking for, pilot?
And but eventually that will lead to the death of the colony. And some of the oldest known colonies. So there's several bamboo corals, as well as several black corals that have been dated to over 4,000 years old. For sponges, that's a whole other question. It's very difficult to study them. So corals have these,
looking down at you.
much like terrestrial trees, they have growth rings. So you can use those growth rings to age them, but in sponges, they have not yet found any kind of structure that could be used to age them. Yeah, so easy. So that's another one of these chrysogorgids near the bottom. That's Pleurogorgia militaris, planar looking fan. And yeah, a lot of small colonies that several of our shorebait scientists have noted.
Come in partial here, video.
Roger, partial.
But yeah, this question of like, where do we find the highest densities? Of course, there's an interplay of many factors. So you need stable substrate. You need a place that these things can grow on without being disturbed by landslide. Then you need favorable currents. And then, of course, these animals need to get here in the first place. So a lot of these seamounts and ridges are very, very isolated. And so getting to them and settling on the first place is a challenge, and in particular in the deep sea, where the so another black coral there on the bottom. Something in a water column just in front of us.
We can try to catch this video.
Roger, go on in.
Looks like a polychelid worm.
Up there.
Alright. Just center them up. Alright,
There you go, coming in.
Alright, let's come on out.
Coming out.
Yeah, hard to say conclusively, but it looked like a polychelid, a polychaete, or also known as bristleworms. Extremely species-rich group, over 10,000 species of these worms.
Pushing ahead.
Oh, and some of our folks at the University of Hawaii Exploration Command Center identified it as an amphipod.
Would you, for when Sirius comes to a stop here, do you want to continue along this ridge up kind of at like 330?
It's, yeah, I don't know. We're gonna have to wait to see where.
Maybe a quick snap zoom on that red animal on the rock.
And wait to see where pilot sees the
Copy that,
ridge
science.
continue.
All right,
Copy
video.
that. Go
Let's
ahead.
let's come in here.
Coming in high.
Copy that, bridge. Thanks.
So that's yeah. So it looks like an ophiuroid on pronoid coral.
And come in more.
So yeah, ophiuroids.
All right, come on in.
Coming in. holding.
Are in a very, very species-rich group that most people are not working on, or very few people are working on, and so this is this is going to be in a group
There, do you have any more?
Okeanos.
Going all the way in. Full zoom.
Nav, when was that ship move? When was that ship move complete? Okay.
Excellent, thank you.
Thanks.
All right, come on up, video.
Coming out. Full wide.
And actually, while we're here, maybe let's take a quick look at that black coral on the bottom.
The black coral?
Yep,
Yeah.
just at bottom.
Camera is down 90 degrees.
All right, I only have a moment here.
Yeah, snap soon, whatever.
All right, come on in, video.
Roger, coming
The
in.
ship is stopped.
All right, copy.
Out there.
Yeah, that's good.
So yeah, that's this Bathy is close to Alternata, and it looks like it has in the center there a pollinoid worm in it. Excellent, thank you.
All right, we can come out.
Coming out, I.
So that's
Back
something
in.
that we saw routinely during the last year's expedition, as most of these black corals that we imaged, and that genus in particular, had a symbiotic pollinoid or scale worm living in the tissues of that central
Now it's time to
portion.
move that
And in those images where we were able to get close-ups, it looks like that pollinoid actually caused modifications in the soft tissue of the black coral, and that's something that had never been described before in the literature. Some nice pillow lavas. Depth is 2,310 meters. So we landed at about 2,360 meters, so about 50 meters shallower and just over 400 meters away from us. So this is a very gradual slope up the ridge. We're really trying to cover some distance and hopefully make our way to a portion of the ridge where there's a high density community.
Alright, coming down. Nav, I think you were right. I think our next one's going to need to be 3, 4, 5. 30 meters.
30 meters at 345.
330. Let's just do it. What you had.
Copy that.
Coming down.
Bridge, this is
35.
Nav. We'd like to request a move of 30 meters at 330 at 0.2 knots.
A few more individuals of these large Farreid sponges. So family Farreidae.
Come back, was that 330 or 23? No, we would like 330.
And that's in the genus Spidoscopulia.
Good copy. Thank you.
Let me get these skids out of the shot, pilot.
Yes, please.
Roger, coming in. It's interesting. I can get the left one out, but the right one's still in.
Yeah.
So glad
Makes me wonder
your
if the
folks at
camera's
the
pointed
University of
center.
Hawaii Exploration Command Center are joining us.
I'm tugging you.
Yes, you are.
Back it up.
like there's a sea star directly
One
under
of
that.
many
Yeah, I was gonna try to
exploration
get to
command
it,
centers
but,
that's operational
no
during this
tether.
expedition.
Oh yeah, you're way out there.
In Hawaii, we have both at the Inouye Regional Center on Ford Island and NOAA offices,
Backing up.
as well as at the University of Hawaii. And we have a few more public viewing displays throughout the state, including at the Maui Ocean Center, the Waikiki
Thank you, Rich.
Aquarium, and the Mokupapapa Discovery Center on the Big Island. So welcome for all of you folks that are maybe watching this on one of the
I
public
got
viewing
something else
displays.
for you
You're
guys.
watching live video footage
check
that's
this.
being broadcast to you in real
Did it just
time
come in, or
from
was it there the whole
NOAA's
time?
I'm tilted
ship
down now.
Okeanos Explorer. We are on an
I've got it
expedition
out of
up in the
frame.
northwestern
So you're
Hawaiian
on a
Islands,
slight
and
partial.
you're watching live video feed being broadcast from a remotely operated vehicle that's diving 1.5
Not yet.
miles deep and about
Just
1,000 miles northwest of
Let's See
Honolulu.
if we can get this sea star from here.
Roger. Going in. Going in slow. Holding this a partial.
Yeah, he's even behind that sponge.
Yeah, so it
Probably. Oh, it's a full zoom.
So that looks like, and I don't know if Chris is following us,
Out of
but that
line.
looks like Henricia,
I
Paparima,
am heading that direction.
and we imaged several of these sponges,
Hold there for wide.
several of these sea stars during a previous expedition,
Yeah,
and
just
a
come
lot
full
of them
wide.
we
I'll
saw on
wait until
sponges.
Sirius catches up.
Roger. Let me get rid of the
Yeah,
skids.
It's the tough part about going downhill.
Might just have to come back uphill.
Partial.
Yeah.
Uphill. Would you like me to follow you?
Yeah. Okay. And tether heave stuff. Let's zoom in on what this guy is in the middle here.
Coming in.
Bouncing around a little bit, but
Holding.
Pilot,
Looks like a
maybe
enemy.
some of these rocks might be loose, some of these angular ones, maybe not the ones that we're zoomed in on right now, but it looks like this might be a good area to kind of take a look around, see if we can go ahead and collect that second sample before we start covering more ground.
Okay to come out.
yeah, come on that video. Yeah, copy that watch lead. See if I can find something around here. You thinking like one of these in here, or?
Do you want me to stop the ship with?
Or maybe just slightly below. I don't know if those look like they might be a little bit cemented. But just trying to keep an eye out for that second sample. That way we can get it out of the way and
Stay right where those lasers are.
go ahead
In a
and
normal
start
routine,
covering ground.
Might maybe would, but he's so
Yeah,
far out
copy
there
that
that
watch lead.
we need Sirius to keep going.
So you're moving more to my starboard.
Yeah, I'm sort of heading towards your starboard, but it'll still give you tether.
I could set down here, watch lead, and see if any of these are loose.
Yeah, that sounds great.
I still have you on a partial.
Yeah, come pull out.
Pull out.
bring up my hydraulics, Bobby, when you get a chance.
Hydraulics are up.
So much tether full.
That's what happens when you go downhill where you're not supposed to.
Thank you, Bridge. Ship is stopped. Move is complete.
I'll try to come down as I can, but we're seeing about 5 meters of heave.
Yeah.
And just as we're setting up to see if we can collect a geological sample, I just updated our position, and so we're about 500 meters from where we started to dive. So we've covered about half of the terrain that we hope to cover on today's dive. Looking ahead, we still have a little over 2 hours remaining of bottom time on this dive. So we hope to secure one more geological sample, cover some ground, and then, of course, we're also, if there's any biologicals, so far we haven't really seen anything that is unusual for the
All
region.
cameras wide.
But we'll keep our eyes open, and I'll ask all my shore-based supporters to also keep an eye on things that are on of substantial significance to collect.
Do you want me to move cameras or wait for your command?
Yeah, wait for my command.
All right.
So these are loose. Might be another loose one up here. But that's just out of my range. Do either of these three look of interest?
The very first one you touched,
This guy?
that's probably, yeah, I think that's a good one.
All right, copy that.
All right, thanks, pilot.
Took the phone back.
That might be a little too big for us right now.
Copy
There's
that.
this angular one there.
Right
bottom
below
of your
us?
screen. Yep,
Yeah.
bottom center of the screen.
Oh yeah, that's also a very nice looking
It might be a
sample.
little big though. It's hard to tell. Prefer if you
Yeah.
didn't continue
Yeah.
going down.
I'm just trying to stabilize myself here.
Understood.
Yeah, that one's looking about 20 centimeters or so in diameter, on the large side, but see what you think when you get stabilized.
Do you want Zeus back up?
My Zeus?
Do you want my Zeus back up?
Well, I'm fine.
Okay.
How do you feel?
I'm fine.
Was this the angular one we were looking at?
Yeah, to the left there, but
Yeah.
it looks a bit big.
Looks a bit big. Okay. So that looks a little big for us, watch lead. We're going to look around a bit more.
Copy that, pilot. Thank you.
All right, you can bring your Zeus back if you want, Bobby.
Okay. I'm right over top of you, so you're fine.
Yeah. We're gonna.
I mean, if
we're gonna
any
look
of the
around a
smaller
little
ones
bit.
there
Yeah.
are happier, but all right, bringing mine up.
All right. Thanks for bringing that up, Bobby.
Brian Kennedy, come back to control room.
How's this guy right in front of us look? Watch lead.
That looks great.
Okay.
It's loose. As long as it's not too big, I think that's perfect for us. Thank you. That looks great.
We have a sample in the port forward.
All right. So if we want to zoom in on this video,
Roger.
You're gonna
We're
want
gonna have
this
to
in
bring
the
your camera up a little bit.
right box, starboard box.
yeah, it's
Coming
copied.
in. Still coming.
So if
Holding
you're just
there.
tuning in, we are diving on
Maybe a
the
wrist
South
rotation while we have it
Ridge
this close.
Rift Zone of Pioneer Bank, about 1,000 miles to the northwest of Honolulu. We are currently in the process of collecting our second geological sample at a depth of
Probably sufficient.
2,314 meters beneath the sea. We are hoping to cover some ground on this dive after we collect this,
Okay.
and
I am full
our
wide.
objective
Okay, full wide, please.
on this dive is to actually try to make some observations
That's
on the
full wide.
extent of the high density coral and sponge communities that have been identified just about a kilometer upslope from us.
Come full wide on.
So far
Full
we have
wide on
seen
All cameras are full
moderate
wide.
densities of sponges and corals, but some beautiful geology of
Yeah,
intact
down a little bit.
pillow lavas and a couple of locations of actual volcanic vents in which were the source of the eruption for these intact lava flows. It was a beautiful job by our
Doesn't look
ROV
like it's
pilots
quite
and team of collecting our second geological
Clear.
sample. Looks to be a nice large sample. It'll provide a lot of work and material for multiple geologists to analyze in the labs later on. Pioneer has actually never been dated using these rocks, so hopefully one of these rocks in the future will
Now,
yield
let's
an
make sure you
absolute
drop a
age,
target, which
the
you
way
already have done.
geologists age
got it.
Perfect.
the volcanoes as we take a sample of rock
Clear on
and
Minisus?
analyze them for the
Thanks, Bobby.
Got it. Bring
Hydraulics
down.
are clear.
radionuclide
hydraulics.
content of the argon actually that is contained within the rock. So
Ramp down.
hopefully sometime in the future that rock will actually help us understand how